Thanks for the invitation to present. So I'm going to tell you a little bit about our unpublished work at this point. And this is really in many ways more of an idea where we're going than a complete story. So, but I do want to show you where we are. If we go to the next slide, please. So, I don't think I need to tell too many people here that detecting cancer early improves survival, and lung cancer is a really good case in point. First, it's one of the biggest cancers in terms of how many people get it and die from it. And depending on how big the tumor is in the lung, when you detect it, your chances of survival change dramatically. So if a nodule is less than 10 millimeters, you have a really good chance of surviving beyond 5 years. But if you're on the other end of the spectrum, and if the nodule is more than 30 millimeters, that drops to less than 60%. And according to the stage, it's the same story. Local disease, 54% 5-year survival. Metastatic disease drops down to 4. We can go to the next slide, please. So the problem, though, is that detecting cancers of any kind at an early age remotely, that is in the blood and through some kind of a liquid biopsy, is really not easy. And the reason is kind of obvious in that if you have a small mass of a tumor, it gets diluted massively, whatever the tumor produces, in the circulation. So there have been some theoretical studies which may be a little unduly pessimistic, but basically based on all the kinetics of secretion, clearance, and sensitivity of various analytical methods, there's been some conclusions that it's difficult to detect primary tumors that are smaller than the size of an olive, which in many cases is a bit on the late side. If we go to the next slide, please. So we at SomaLogic have been working with a multiplex proteomics method called Soma Scan, which takes a collection of modified aptamers, they're DNA based. And we have developed a way that we begin with incubating, at this point, 7,000 different somomers, as we call them, with a mixture of proteins. These modified aptamers will find their proteins, form stable complexes. We remove everything that's not bound. And at the end of the assay, we measure only the aptamers themselves, viewing them at this point as if they were simple single-stranded nucleic acids. And so we have translated basically the amount of protein into measurement of the amount of DNA that's left at the end. Next slide, please. So one example that, just a visual if you want, we measure 7,000 proteins in some baseline state. If you click to the next, then we measure the same group of proteins after something has changed. There's been some kind of a perturbation. And then the various proteins that we measure can be grouped into categories. If you can click to the next one, so that they indicate, and the next one, please, they indicate the various conditions that you can measure. So these we call Soma signal tests. We go to next slide now, please. We have applied this to lung cancer, where we have, you might need to click a couple of more times. To a couple of conditions which are shown on this slide. I'm not going to go into detail here, but basically the idea is that from one and the same blood sample, you can detect multiple conditions by looking at protein changes by measuring all 7,000 proteins that we can, putting them into different groups. And so coming back to lung cancer, one study we did a few years ago involved resections of non-small cell lung carcinoma from patients, and then taking samples from the tumor tissue itself and comparing it to the proteomic composition at the uninvolved normal lung tissue. And what we see is that some proteins go up, some proteins go down. And taking one example here of thrombosponin 2, we see it dramatically elevated in the lung cancer tissue itself. And of course, in this kind of experiment, you take a tissue and you grind it up and you kind of lose track of where all the proteins are in that tissue. But after the fact, you can take one and the same affinity reagent and you can stain the tissue to find out where that original sample was, which in this case is in a tumor stroma, but is absent in a normal tissue. So if we go to the next slide, so detecting protein changes in the tissue itself is in some ways much easier because the changes are bigger than detecting the same things in blood. And we have shown this in a study where we actually compared a Soma scanner experiment run in plasma or serum rather, with what happens in the tissue itself. And what you see on the slide is that much bigger changes are observed in the tissue itself. Can you go? No, you can, yeah. So for this xenograph study, and this is where I begin with data that's not been published, we chose two different cell lines, and they secrete what they want to secrete in just the culture. And then we implanted the same tumor cell lines in mice. And we then simply measured the proteins that we can measure in plasma. And if you can just go to the next slide, please. And these tumor cell lines are erlotinib sensitive and resistant. We implanted a typical 10^7 cells. And if you go to the next slide, what we observed is that these tumor cells in mice now grow at somewhat different, but basically similar rates. If you go to the next slide, please. So if you look at these volcano plots, what you see is that changes in tumor profile of proteins that we can measure in plasma as the tumors grow is really rather massive. The way these volcano plots work is if you plot on the x-axis the fold change of any given protein on a log scale, log 2 scale. And then you look at statistical significance on the y-axis. Things that are beyond these bands basically give you significant signals. And so compared to controls, which don't have any tumors, there are some changes because these mice actually grow over time. But you can see that in mice that have tumors, these are massive changes in protein compositions where most of them actually go up in concentration in plasma. There are some that go down, but as you would expect, these tumors that secrete proteins that the mouse really doesn't see in under normal circumstances are dramatically elevated. So one of the questions that we can ask is, if you go to the next slide, please, is what, this next slide is a little bit slow to load. Sorry. No, sorry. If you go back to the one that is kind of a heat map looking thing. Sorry. It's nice to have something to drive for you, but sometimes it I'm not seeing it on the screen that everybody else can see, so I'm just gonna. So what the next slide shows is I mean, it's kind of the same as the one before, which is if you look at the changes now, the way to read this, you go from top to bottom, day 0 to day 40 in tumor-bearing mice on the top half of this plot compared to naive mice that don't have any tumors. And so as a certain protein goes up, which are all shown on the x-axis, and these are for significant proteins that change with below a false discovery rate, statistical significance of 0.05, and that change at least threefold. You can see that there's a massive number of proteins that changes in these mice to have the tumors compared to controls. And so the question that comes up was, well, what are these proteins? And so we wanted to ask a fundamental question. As these tumors grow in mice and secrete tumors, what can you see first? If we go to the next slide now, please, for these two different cell lines, again, they're both non-small cell lung cancer cell lines. If you look at the far left set of panels, these are values in plasma as a function of just time. And the two different cell lines are either in red for the erlotinib sensitive or in blue for the erlotinib resistant cells. For these three particular markers, they both go up in both tumor types. You can plot these in a slightly different way, and you can ask, well, what are the changes according to tumor volume, which may be more interesting than time. You can see again, they both go up. Now, you can see the same proteins being expressed in the cell lines themselves in culture. So these cells are capable of producing these proteins, and you can see them in plasma of these tumor-bearing mice. You can also take the cells themselves, lyse them, and ask the same question with the same result, or the tumor that you extract from the mouse at the end of the experiment. Now, without going into the identity or the importance of these proteins that we see, I will draw your attention to one of them, which is GAPDH. GAPDH, many of you use as a control for a housekeeping protein that is present in all cells. And it's probably one of the most boring things you can imagine. And yet here it is among one of the first proteins that you can pick up in plasma of mice that have these tumors. So obviously GAPDH has an important role in metabolism. And this is one of the things that we see. So in the next two slides, what we have put together is just evidence that there are, on the other hand, while there are some common proteins, there are some proteins that are unique to either one or the other cell type. So tumors, of course, are different from each other, and each person will idiosyncratically have a tumor that is different from any other tumor, and yet there are things that they share. So if we go to the next slide, you'll see some markers that are unique to this erlotinib-resistant cells. Now, we decided to expand this experiment to include, on the next slide, please. Oh, sorry, before I go there, we then took some of the proteins that you can detect early. We developed a model based on measurement of multiple proteins at the same time. And what we have observed is in the top two panels, you can use markers that are common between these two cell lines, unique for one in the middle, and then for the other on the bottom, to develop a prediction model that allows you to identify these tumors at an early stage. And you can see that we are getting to a point where we can detect tumors that are in many cases well below 1 cubic millimeter. Now, of course, you have to remember this is in mice. And so a 1 millimeter tumor in the mouse is a whole lot, bigger if it was in a human, but still, this is a starting point for identifying such proteins that could be important early biomarkers. If we go to the next slide, please. Now, we expanded the study to go from lung cancer to 3 other cancer types: ovarian, colon, breast. And you can see on the bottom left graph that these tumors grow at different rates, with the breast cancer 2 cell lines that we chose growing at the slowest rate. So we adjusted the time frame of the sample collection according to tumor growth. And what we then did on the right-hand side is basically a summary of a very important observation, which may not be all that surprising to many of you, which is like what we have seen in lung cancer alone, where we could differentiate the 2 tumor cell lines, but also find things that are common. We can do the same with the other 3, or really all 4 together tumor types. We can find unique between 2 cells, common between 2 cells, and then we were also able to identify 15 common markers that all of these tumors share. And again, this is based on our ability to identify these proteins in plasma at an early stage. And developed a model for this common model where, it's the 15 biomarkers that all of these 4 tumor types share with reasonable detection analytics. So that's basically where we're headed. If you go to the next slide, please. I'm going to say a few words about where we are headed from here. So now this is coming all the way back to non-small cell lung cancer. The 2 cell lines that I've described are erlotinib sensitive, erlotinib resistant. And so now you can ask a question: what happens to plasma biomarkers in mice that are carrying erlotinib resistant cells, which are shown in blue here, which grow a little bit faster? And when you treat these mice with erlotinib, the tumors obviously continue to grow because they're resistant to the drug. But what are the changes in the proteome anyway, which may indicate toxicity of the drug without any anti-tumor activity? On the other hand, you can take the sensitive cell line, you can treat these mice with the drug, and you see the tumors growth being attenuated. And then you can ask, what is the protein profile in plasma at that point, including as you treat these mice multiple times, as of course humans are treated all the time. So this is the direction we're headed next. If we go to the next slide, please. And so this, in a way, summarizes the experiments that we have done. We've developed a way of measuring changes in protein plasma in mice that have human tumors growing in them. Everybody appreciates the limitations of these xenograft models. These are human cell lines growing in mice. These are not real tumors. But what this model does allow us to do is to monitor changes early after tumor implantation, which is very difficult to do in another setting. So we found both unique and common markers for all 4 different tumor types we have examined. And we found that we can develop tests that are based on measurement of multiple proteins at the same time that allow us to identify these tumors at an early stage. And I'm looking forward to telling you a little bit more about where this goes in terms of exactly the composition of the proteins that we're seeing, other than GAPDH. And thank you for your attention.